Dear students, welcome to the EPG Pathshala. I am Dr. Rajkishore Sharma from Chemistry Department of Delhi University. Today, we are going to discuss the module Analysis of Secondary Ion Mass Spectroscopy. This is under the paper of Surface Analytical Techniques 2. As you see, the name suggests analysis means the detailed examination of the element or structure of something. Similarly, in this module, we will study how to interpret the data recorded in a SIMS experiment. Now, let us see the overview of this module. In this, we will be going through the following topics: dynamic secondary ion mass spectrometry, the time of flight, vacuum, conditions of analysis, primary beam, beam angle, angle of incidence, sputtering rate. We will also discuss the detected area, species monitored in SIMS experiment, energy distribution, the insulator effect, analytical information through SIMS experiment, sample preparation, data interpretation, and finally the advantages and disadvantages of the SIMS. As you know that among all the microbeam techniques available nowadays, secondary ion mass spectroscopy, or popularly known as SIMS, is most versatile. By combining in situ microbeam measurements, while the high specificity and sensitivity of mass spectrometry, the SIMS has capability to detect most of the elements in the periodic table with a high dynamic range. SIMS is also a well-established technique in semiconductor industry, like it is used to measure the concentration of dopant elements. SIMS have also been employed to determine trace elements in minerals at concentrations that are very low and to be detected by various other methods like electron microprobe analysis, which is known as EPMA.
Which again deals with the same phenomenon of secondary ion mass spectroscopy. The way this analysis is carried out has been explained in this slide. SIMS is highly effective technique which is used in the analysis of stable and radiogenic isotopes in minerals. Other techniques are also there, but this is one of the best techniques for doing this. Analysis of SIMS can be done broadly either from dynamic secondary ionic mass spectrometry. In abbreviated form, we write it as DSIMS, or TOF, SIMS, which is in the expanded form would be called time of flight secondary ion mass spectrometry. In dynamic SIMS, the number of ions that are incidents exceeds the number of atoms present on the surface, resulting in the elemental information having high count rates, thus allowing trace analysis also. On the other hand, static SIMS is used to acquire molecular information also. There are two forms. It talks about one of the forms mentioned in the earlier slide. This is dynamic secondary ion mass spectrometry. In brief, as I said, DSIMS is the way it is abbreviated. Salient analytical feature of DSIMS are it monitors elemental composition with very low value up to the level of parts per million or parts per billion sensitivity that allows ready identification of dopants, impurities, surface treatments, and process contaminants. In this technique, we can find out very precise information about dopants and impurities, which otherwise would become extremely difficult to find out. Then we go over to depth profiling, chemical mapping, reflective 3D analysis, etc. The depth profiling helps us in analyzing chemical maps area, selected depth profiles from complex materials. What way selected map area provides various kinds of species on the surface can be found out with the help of depth profiles of the material. The cross-sectional analysis gives information regarding a structure that are effective on complex or thick layer materials. They are highly used in quantitative depth profiling of impurities in semiconductor substrates, etc., or analysis of compositions of glass layer structures and in-depth investigation of the biological surfaces. And information about the second kind of SIMS, which deals with time of flight information. So in this slide, we talk about time of flight secondary ion mass spectrometry. Salient analytical feature of time of flight SIMS are it gives information regarding elements and molecules that constitute the uppermost 1-2 nanometer of the surface. Through the use of the extensive library spectra and accurate mass analysis, we can get detailed chemical information with the help of the second form of SIMS. It talks about detection sensitivities, which again are very low, that is in the range of parts per million for most of the elements. It also talks about chemical imaging of elements and their isotopes and molecular species, along with some micron lateral resolution. Time of flight SIMS is widely used in the analysis of surface additives on polymer material, observation of stains and rinse residue on semiconductor wafers and devices. Chemical imaging of additives and contaminants on polymer films can be very conveniently obtained with the help of time of flight SIMS. The working of time of flight secondary ion mass spectrometry has been illustrated. Here it talks about what way a primary ion beam in the region of 1 to 50 kilo electron volts is focused on the sample and what way time of flight measurements of secondary ions which are liberated is carried out. Emissions of neutrons, positive or negative, secondary ions will emerge from here and are measured.
Now, let us discuss about vacuum. Vacuum is an important requirement for the analysis as the instrument is kept under ultra-high vacuum. The level of vacuum in SIMS is the highest among all the analytical instruments and require utmost care along with sample preparation and the maintenance of the equipment. In the analysis chamber, the vacuum level gets down to 5 raised to the power 10 minus 10 torr, which is equivalent to about 1010 molecules per liter. So it is a very low concentration. While air has 2.7 into 10 raised to the power 22 molecules per liter. Under these vacuum conditions, the possibility of a secondary ion hitting a gas molecule within the instrument is almost zero. It is important requirement for the analysis as the instrument is kept under ultra high vacuum conditions.
The conditions of analysis. If one has to use SIMS in either form, there are certain conditions which have to be followed. The various conditions that are needed for this are primary beam, then what is the beam energy, angle of incidence, sputtering rate, detected area, species monitored, energy distribution, and the insulator effect. because insulator will not allow normal phenomenon to occur. So what way this effect is also included here. Primary beam. What is primary beam and what are the ways it is put are given here. It is subjected on primary species which depends on various factors like detection limit, depth resolution, charge neutralization, microtopography, etc. Commonly used primary beam species are O2 plus, CS plus, xenon plus, and gallium plus. In order to operate efficiently, one has to sacrifice some sensitivity or detectability. If you operate more efficiently, one will find that the sensitivity or detectability limit goes down. And if the limit has to be kept at a very, very low level, then maybe the operating efficiency has got to be compromised. Then the most important factor, which is about beam energy, is given the characteristic feature of beam energy. The type of beam energy has direct impact on depth resolution, secondary ion yield, and sputtering yield. With high energy, primary ion current and focusing are improved. How high energy in primary ion current is used, that would improve the focus.
Now about the beam. The beam that has to be focused on the sample has to be at the angle for proper sputtering. Now about angle of incidence. The yield of sputtering increases with rate of incidence up to a certain angle and then decreases. In case of multi-layer structure, 60-degree angle of incidence provides approximately equal sputtering yield. As the angle of incidence increases, the yield of SIMS, the yield of secondary ion decreases. The angle of incidence is affected by the potential of magnetic sector.
Area of detection. In case of depth profile, there is a contribution from the crater wall as a result of total rastered area. This can't be used for detection. When primary beam is at the center of the crater, a large raster to detected area is required for the microphone mode. This talks about sputtering rate and energy distribution. Sputtering rate will talk about the rate of sputtering, which depends on various factors: the energy, angle of incidence, the mass of primary angle, the mass of sample atoms, the binding energy of the sample, current density of the beam, which is equivalent to beam current upon rested area. Then energy distribution. Depending upon what way the sputtering rate is analyzed, energy distribution would be found out. Since molecular ions have a narrow energy distribution than atomic ions, in such cases, high pass energy window is chosen in order to select atomic ions.
Next is the monitored species. In case of species monitoring, the various factors that must be kept, there are various factors that must be kept in mind. These are the detection limit, the range in which species can be detected, sensitivity versus dynamic range. The detection limit is influenced by the following factors. These are the low count rate, high implant standard dose, contaminated instrument, whether it is poor vacuum or contaminated primary ion beam.
Shall talk about insulator effects, which is one of the important factors while talking about the operation of SIMS. The distribution of energy can be shifted in case of charging of insulator during analysis is concerned.
Now about analytical information. Analytical information refers to the information that is provided while we are analyzing the sample. These are as follows: the mass spectrum, which identifies the composition of the element and ion or ion of the uppermost surface, which is approximately 10 to 20 angstroms of the analyzed surface, from positive and negative mass spectra. Number 2 is the depth profile. In this, continuous analysis helps in obtaining composition information as a function of depth. Depth resolution is possible for few angstrom. High sensitivity mass spectra can be generated at any depth of the profile. Now, the third point is secondary ion mapping. It measures the lateral distribution of element and molecules on the surface of the sample. In order to obtain a SIMS map, highly focused primary ion beam is scanned in a raster pattern across the surface of the sample, and the secondary ions are analyzed on the selected surface area. The sample that has to be analyzed also require very accurate preparation for the best possible results. In SIMS, sample size should not exceed 3.5 inches in any lateral direction. While preparing sample, one must keep the following points in mind. These are the height. The height should be up to 0.8 inches or about 20 millimeter. The sample must be stable under ultra high vacuum or UHV conditions. UHV condition are important in order to prevent contamination of the sample. During analysis, SIMS technique is restricted to the surface and can detect species that are present in the concentration in the range from ppm to ppb. It must contain minimum amount of water and salt.
Sample preparation is one of the very important requirements when analysis through SIMS is carried out. Samples to be analyzed include powder bulk films, fiber particles, thin films, spin cast onto substrates. The thickness and morphology are very different as they will affect the probability of generating secondary ions and the stability of SIMS spectrum. The surface of the sample must be clean, as any contamination on the surface during handling or processing can affect the accuracy of the analysis. As soon as the sample is prepared, it must be analyzed in order to get rid of the various difficulties like surface diffusion, molecular reorientation, or surface contamination by the lab environment.
Detection limits and sample degradation. Detection limits for most trace elements are between 10 raised power 12 and 10 raised power 16 atoms per cubic centimeter. This depends on the type of instrumentation used, like the primary ion beam used and the analytical area. It depends on some other factors as well. Samples as small as individual pollen grains and microfossils can yield results by this technique. Amount of surface scattering created by the process depends on the current, that is the pulse or the continuous mode, and dimensions of the primary ion beam, while only charged secondary ions emitted from the material surface through the sputtering process are used to analyze the chemical composition of the material. This represents a small fraction of the particles emitted from the sample. Now, we will talk how SIMS works. There are several different designs of SIMS currently being manufactured commercially that have applications in the geosciences. Most of these instruments are characterized by a source region in which the intensity, energy, and orientation of the primary beam, orientation is relative to the sample, orientation of the primary beam are controlled. Ions generated by this process from the secondary beam and are subsequently transmitted within a continuous high vacuum environment to a mass spectrometer. Most SIMS instruments used for elemental and isotopic analysis function by accelerating ions produced in the source along a potential gradient, typically 10 kilovolt, and then transferring these ions into the mass spectrometer. Details of the configuration of the mass spectrometers vary from one application to another, but all utilize both magnetic and electrostatic analyzers, commonly referred to as the sectors. If the electrostatic analyzer or sector precedes the magnetic sector, the design is referred to as forward geometry. The advantage of this configuration is that the electrostatic filter reduces the energy range of the secondary ions so that they can then be separated into independent ion beams. This is based on the charge and mass ratio, independent of ion beams, by passing them through a magnetic field or a magnetic sector. In this configuration, multiple ion beams can be measured simultaneously. If magnetic sector precedes the electrostatic sector, this is called the reverse geometry. Then the mass resolution in reverse geometry is improved at the cost of losing the ability to measure multiple ion beams simultaneously. Other types of mass spectrometers can be coupled to the SIMS source, including quadrupole and time of flight analyzer. These later configurations have fewer applications in the geosciences. Isotope ratio measurements. Isotope ratio measurements are operationally similar to depth profiles, excepting that precision and accuracy requirements are higher. Since all of the isotopes of an element have the same chemical properties, ionization and detection efficiencies remain nearly constant for the different isotopes. Precisions of 0.1 are commonplace and accuracies approaches precisions. Error analysis indicate that the precision are limited mainly by Poisson counting statistics. To attain these accuracies, SIMS instrument must be carefully tuned and interferences must be eliminated. Mass spectral peaks should have flat tops, steep sides, so that the slight magnet instability does not change the ion signal intensity. The figure shows the necessary flat top peak and the high mass resolution used for elimination of oxygen interferences at m by z 32. Both are necessary for measuring accurate 34s/32s isotope ratios. The ratio of isotope signal intensity must be corrected. The ratio of isotope signal intensity must be corrected for slight variation in detection efficiency at different masses, and for slight variations that depends on signal intensity. These corrections are usually larger than the range of expected isotope ratios.
Data interpretation. Data interpretation is done by taking examples of organic materials. There are three spectral regions that provide information regarding their surface area. The three regions are as follows. Sub-mono mass region. It provides information on the chemical structure of the constituents. The next region is N-mer-mass region. It contains information regarding polymer structure. Third region, about which we've been talking about, is oligomer mass region. It provides information regarding the average molecular weight distribution of the concerned polymer. It is not possible to get quantitative value like degree of unsaturation, but it is done by comparing the data with the spectra of known reference sample. The various values depicted are as follows. Unsaturation. Every polymer contains unsaturation, and the relative degree of unsaturation is calculated from the yield of hydrocarbon secondary ion within a CnHX positive cluster series. Then we have aromaticity as one of the important regions about the information is given. By visualizing the spectra, the presence of aromatic groups is evident by the presence of secondary ion with aromatic character. Branching or cross-linking can be analyzed with this. By taking the total secondary ion yield, the cross-linked character of the polymer can be investigated with this. The probability of emission of secondary ion depends on the environment, and with the increase in the cross-linked character of the polymer, there is a decrease in the total secondary ion yield in such cases. Surface functional groups. Various polymer classes contains a backbone of hydrocarbon with a functional group comprising of the side chain. The general structure of these polymers is CH2CHX with n number of units, where X represents the side chain having functional group. Data interpretation. During analysis, we cannot solely depend on fragmentation and reorganization reaction, as assessment of financial group is difficult, in which the sample contains various functional groups like plasma polymers or plasma modified polymers. By relative information about the different properties of samples, that is degree of unsaturation, this is obtained by comparing the different SIMS data.
We will talk about the advantages of SIMS, secondary ion mass spectrophotometer. The advantages are as follows. The elements from hydrogen to uranium may be detected. Most elements may be detected down to concentrations of 1 ppm to 1 ppb. So this talks about the sensitivity of it. You can detect concentration as low as 1 ppb. The isotopic ratios may be measured normally to a precision of 0.5 to 0.05%. So it has a high precision, and the range is from 0.5 to 0.05%. Two-dimensional ion images may be acquired. A secondary ion leaves the surface at a point close to its original location. This enables localized analysis of the sample to be undertaken and is the cornerstone of ion imaging. So this is treated as cornerstone of ion imaging. The volume of material sputtered is small. Using high energy and high primary beam densities, that is the dynamic SIMS, a volume of 100 to 1000 micrometer cube is analyzed. In contrast, using low energy and low primary beam densities, static SIMS. This is the static SIMS. In contrast, using low energy and low primary beam densities in static SIMS, the material sputtered is exceedingly small, with surface monolayers lasting hours or days. And the third point is three-dimensional ion images may be acquired. 3D ion images may be acquired by scanning, or you can call rastering, the primary beam and detecting the ion signal as the sample is gradually eroded. Little or no sample preparation may be needed is again an added advantage of the SIMS. So sample preparation is extremely simple for SIMS analysis. Now we will talk about the limitations of SIMS. The material sputtered from the sample surface consists not only of monoatomic ions but molecular species that in places can dominate the mass spectrum. So at places this can dominate the mass spectrum, making analysis of some elements impossible. So this thing makes analysis of certain materials impossible. The second limitation is the sputtering process is poorly understood. No quantitative model, the sputtering process is poorly understood. No quantitative model currently exists that can accurately predict the secondary ionization process. So accurate prediction of the secondary ionization process is difficult. In order to obtain quantitative information, a suitable standard has to be used and empirical corrections are to be applied. So here we conclude by telling you about the advantages and disadvantages of SIMS. The advantages of SIMS are as follows. SIMS can distinguish elements, those can be detected and are isotope of the elements. Detection limit of most element is ppm and ppb for most of favorable elements. In case of SIMS, there is good depth resolution and good lateral resolution. For SIMS, we can obtain chemical information from relative molecular abundance. Last but not the least, we will discuss about the disadvantages of SIMS. Disadvantages: there is a lot of mass interference. The yield of secondary ion varies by more than 6 orders of magnitude over the periodic table. The yield of secondary ion depends on the matrix. In order to quantify data, numerous secondary standards are required. To obtain the best depth resolution, flat surface of the sample is required. So these were the disadvantages of SIMS technique. Thanking you.